Hello, good evening. Hi there, welcome to the Wayne Theater. My name is Kim Ryder, and I am the Director of Operations here at the Wayne. And on behalf of our staff, our ambassadors, and our board of directors, we welcome you here this evening. Thanks so much for being here. And we love having these science talks here for you all and for us. It's just so fun, and we love to see our cadets from Fishburne joining us too. So we have wonderful events coming up. We've got Daly and Vincent coming later this weekend. We have the Kingston Trio, but we also have the Spirit of Scotland later on in October, as well as the Tanzanite African Acrobats. All those are going to be just so cool. It's going to be a journey around the world coming here at the Wayne for the next couple of weeks. So it's really exciting things that are coming up. We also have a production of The Woman in Black. That's going to be right around Halloween time, and that's going to be a fantastic production. We're doing that at the Wellington Barn, the Wellington venue, so we're partnering with them to do a production, so we're very excited about that as well. So without further ado, again, thank you all very much for being here this evening, and I'm going to turn things over to Mr. Joe Kuiper.
Good evening, everybody. I'm Joe Kuiper. I'm director of the Virginia Museum of Natural History. I can't see anybody. First of all, I want to welcome you. Thank you so much, and thanks for the Wayne Theater. This has been a long-standing partnership between the theater and the Natural History Museum, and I just couldn't be more proud to continuously be here year after year with very quality programs. And I also want to thank everybody who's joining us online, or if you join us at a later date, this is going to be recorded and archived. So that is awesome. I also want to thank the South River Watershed Coalition, been an important partner in everything we're doing here. They have also sponsored this lecture series, and we just couldn't do this without them. So thank you very much. Please, at the conclusion of the talk, stop by their display table out front, and you can see some examples of the work that they are conducting, as well as some of their outreach materials. It's quite fascinating. So we love to see them here as well. So thank you. And Max, thank you. Max Quillen will be out there manning that table. So please come by and say hello to him. So before we get to our talk tonight, of course, we have gone through like the last 2 or 3 years trying to get a branch campus here. Maybe it's been a little longer than that. Right? I think it's probably been over a decade. In fact, it has. But you know what? I want to thank you for your patience with this. These are never short-term projects. If you look at the building that we live in down in Martinsville today, that was opened in 2007, and I started in 2010. So according to the people who've been working there longer than me, it was over a decade from the time they first started really pushing the idea of we need to move from the renovated school building that the museum was born in the 1980s to a modern facility that can care for our collections, which occurred. We are now asking the state to promote a branch to bring our resources in education and collections management to the Shenandoah Valley. Why? Because one, you have such fascinating stories to tell here. We can tell local stories about the natural history of Virginia right here. Draw a 50-mile radius around Waynesboro, and you have a plethora of excellent stories to tell that tie right into everything from science SOLs to current research being done right here in the Commonwealth. The second thing is that you have a much different population than we have down in Southern Virginia, mainly because you have Shenandoah Valley, which is very popular, but you also have Skyline Drive and Blue Ridge Parkway that also intersect with the Appalachian Trail. So, I mean, there are just incredible resources here. What a perfect place to put a branch. The efforts that you all have put in so far, I will tell you that talking to our local delegate and senator, talking to the Secretary of Education, they specifically have all told me, Joe, we have never seen as many letters of support for a project than for this. So you all have done your part, and thank you so much for that. That's what it takes. We've got one more step to take. We received permission from the Secretary, and that means from a collaboration of different folks in higher government than we're at, and they gave us the approval to make the budget proposal for moving into the capital construction pool. So to back up, we've been in the 6-year capital outlay plan for the Commonwealth. The next step is to matriculate to the capital construction pool. We've now been given permission to apply for that movement, and as such, we have that last step. So we are hoping that we will be within Governor Youngkin's proposed budget for the next biennium. That will come out in December for consideration, and then of course debated in the next General Assembly session, which will be overseen by a new government. We of course are going through a very significant election cycle here. All that said, it's always advantageous to be in the governor's budget regardless of what happens. So all you have to do now, this is it, this is the last thing you got to do, send the governor a quick note. You can do it online, you can leave a phone message, even better, send a stamped envelope to the governor's office with a message of your support to bring the branch of the Virginia Museum of Natural History right here to downtown Waynesboro to service this area of the Commonwealth, as well as all the tourists traveling Blue Ridge Parkway and Skyline Drive. So that's where we stand. The only reason we're here is because of you. I don't mean that. I'm not trying to placate anyone. I'm being honest. We would just be another state agency hoping to grow. That's the way they always are. But to hear the voices of the citizens is incredibly significant. So speaking of the resources and the stories and stuff, I'm very proud to be here today because I brought one of my colleagues that I get to work with at the Natural History Museum. Marshall Boyd, it does say research and collections technician. I kind of consider that a title that we give people, but really he is a Renaissance man as far as the sciences go. We spent the day joyfully walking around Maple Flats, I don't know, a dozen or so miles south of here. I got introduced to marbled salamanders and saw some spotted newts, as well as some grass frogs and maybe a few other things. So it was a beautiful rainy day for walking around being schooled by a herpetologist, but he does more than that. Marshall comes to us with a degree from Salisbury State in Maryland. He also has experience working as a collections manager at the Smithsonian. And so we're very glad to have been able to recruit him for the last couple of 3 years. I've lost track, right? And he's been with us. He's now part of the museum crew, really working hard in the herpetology collections, but he also has done work in the fish and the mammal collections, amongst others. And that's what makes him a Renaissance man, to be able to jump taxa like that, to jump different groups of organisms, and he does it very well. And today he's going to do kind of a cool follow-up. You may have seen Dr. Ariana Kuhn's talk. She is our former curator of herpetology. She just took earlier this year a plum position at the Illinois Natural History Survey. We are so proud of her. That is an incredibly venerable and respected institution. I don't blame them for recruiting her. So that position is vacated, but in the meantime, she did do a talk here at the Wayne Theater last year. Marshall's going to do a follow-up about taxonomy and how our collections contribute to this field of systematics and taxonomy. So he's going to give a little different take on it from his own perspective. So without further ado, let's please give a warm welcome to Marshall Boyd of the Virginia Museum of Natural History. All right, man, knock him out.
Thanks. Hi, everybody. I am going to forewarn you that I do not stand still. I talk a lot with my hands, and so I will probably be all over the stage. So hopefully everybody's watching at home pretty well. Thank you, Joe, for that wonderful introduction. I've quite enjoyed my time so far at Virginia Museum of Natural History. Within research and collections, like Joe was saying, I've gotten the chance to work with all different types of collections. You guys are going to see some pictures that we have from behind the scenes. We have a plethora of collections that across all of our taxonomic groups, and I get to work with everything that's vertebrate zoology. So birds, mammals, like Joe said, the fish. Within herpetology, we have reptiles and amphibians. We also have a nest and egg collection related to birds that I'll show you guys at least one picture of that's in here. But my passion has always been for biology since the beginning. Since I was a little kid, I've always kind of been that outdoorsy kid that would go out. I was in Boy Scouts, received my Eagle Scout years ago, and just always kind of been going around finding things out in the woods, including this beautiful animal here. This is a timber rattlesnake. I'll get into the details of her in just a little bit. Obviously, as you can see, was not very excited to see me, but I kept a safe distance because of the fact that as a herpetology person, I am passionate to be able to go out and see these animals, get nice photos, beautiful videos. I actually have a video that I should have included of her rattling right at me, but I made sure that I was at a safe distance because of the fact that it's something that I want to be able to show you guys that these animals are not just scary. So the title of my talk is What's in a Name? The original plan that I had with this is related to the Shakespearean quote of what is in a name, about any herp that would be found as just as beautiful as the next. But we wanted to go into the overall of how taxonomy fuels the museum world. So we're actually going to go on a bit of a journey together to kind of look at what you would do to name a new species. There's actually, at the end of the talk, I have a few species that have been named over just the last couple of years, and it kind of is a continuously growing science that never really ends, and that's part of biology. It's a constant understanding that we are still learning as we go, and just because we've learned it doesn't mean we can't relearn it. We can't learn a new version of that and be able to kind of include that for the next generation of scientists and kind of inspire them. So a lot of what I do is a lot of outreach and collections work, so we're going to jump right into that. So there we go. Just to kind of give you an idea of some of our team, a few of these people you've met before. Dr. Ariana Kuhn, as Dr. Kuiper had said, has moved on to the Illinois State Survey. She and I are still collaborating on our work together. I will actually see her next week. I'm very excited. We have our newest visiting scholar, Dr. Colin Van Buren. He will probably be giving a talk at some point soon, when Joe finally strong-arms him into it. Dr. Cal Avanov in the middle there, as well as Dr. Jackson Means, have also given talks here at the Wayne Theater, looking into all sorts of taxonomy. And then Liberty Hightower, who is a technician opposite me within the entomology and recent invertebrates department, hasn't given a talk yet, but we work very closely together for our collections. But everybody within this whole group has a passion for taxonomy, has a passion for biology, wants to know more about it, and is always curious about, like, what is the next thing we could learn about, but also kind of looking back behind us and making sure that just because we've learned a lot about it already doesn't mean we can't learn more, including Liberty. I'm actually going to give her a very direct shout out to, because of the fact that she's used our collections to understand the taxonomy, or not, excuse me, the taxonomy, the anatomy of squirrels, and being able to look at the fatal aspects of a fall of a squirrel just based on our postcranial skeleton collections that we have at the Virginia Museum of Natural History. Postcranial being everything beyond the head. She looked at leg bones and saw that there was a lot of things that you could see, even though we already had those collections for a long time. So it's constantly growing everywhere we can find. So we're going to look into what is in a name specifically. You are here doing your homework. Everybody do your homework. A very important aspect of the academic side of this. Looking at field work. Checking collections, which never seems to end. What's in a record. Different types of preservation, so what do we have in our collections behind the scenes, what specific types of collections we might have around the museum. Sharing of knowledge, which is pivotal to the overall world of science, and then ultimately publishing that so that way people like you can understand, the general public can get an idea of what we work on behind the scenes and where we can use that research to help out the rest of the world. So starting with our beginner right here, we have our timber rattlesnake, which is Crotalus horridus. Horridus sounds like a very horrible word, in my opinion. Considering it's such a beautiful snake, Crotalus comes from the word crotalum, which means rattle, and then horridus means dreadful, so it is dreadful rattle. So usually when you come close enough to a rattlesnake, it is usually the rattle and then potentially the bite, but most of the time it's a rattle and then the snake kind of slithers away. This animal, although it was very close to me, had no interest in biting, was very much just like, I'm going to sit here and tell you to go away because I don't want you near me. And if you do come near me, then I will have to defend myself. No snake will actually come after you. No snake is actually out there to pursue you. It is only to be there because it wants to go find a mouse. It wants to go find a frog. It wants to eat that. And its only interest in potentially trying to bite you is out of fully in defense. She was really pretty. I was very happy about that. I love that snake. Ironically, I found this with our mammalogy curator. You always find things you're not looking for when you aren't. You always find things you're looking for when you're not looking for it. For example, when Dr. Kuiper and I were out today, I was hoping to find a snake. Wasn't the greatest conditions because it was cooler and about to rain, but every time I'm looking for a snake, I never find it. I have to give a shout out to Carl Linnaeus. Everybody has probably heard of Carl Linnaeus before. Lived from 1707 to 1778, born in Sweden. Carl Linnaeus was noted for the formalization of the binomial nomenclature, which we'll get into the details of binomial nomenclature, but overall the genus species, the two-name system that we use today to understand biology, and the naming of animals and plants, everything that we have in order to create our taxonomic system. It's Latin-based, mostly. Sometimes it's not perfect because of the fact that either other languages get involved or names get involved, but we'll get into that. And ultimately, it has been used within the Latin system because of the fact that Latin is not changing. Latin is considered a dead language, which usually kind of gives the wrong idea. Latin is not spoken commonly, but it is studied. I have actually a really good friend who studied Latin that knows the language, understands how the words work, but Latin is considered a dead language because of the fact that it's no longer changing. It's no longer being built. The changing of the spelling doesn't really seem to happen, doesn't happen anymore. So it's considered not necessarily dead, but kind of stagnant. It stays the same, and therefore it makes a good system for allowing for the kind of sharing of knowledge across the world to understand what things are. But we can't talk about Linnaeus without going into Bauhin. Gaspard Bauhin lived long before Carl Linnaeus, was born in Switzerland, was actually one of the original people to describe a lot of plant species. He was a well-known botanist. He described thousands of species of plants, and then most of those were adopted and honored by Linnaeus later on in life, about 200 years later almost, and overall was included in a lot of what we now know with a lot of plant taxonomy. And Bauhin was actually honored with the Bauhinia genus, which is a genus of flowering plants. Overall, and then later on was describing the Bauhin's valve, or the ileocecal valve, which is actually the valve about down here where your small intestine meets your large intestine. He was also involved in a lot of anatomy. So this guy had a lot of involvement with the overall taxonomy known nowadays, but Carl Linnaeus usually kind of gets the kind of fame for it. So, what is in a name? We mentioned the timber rattlesnake, and just to be fair to our other two species of venomous snakes that we have here in Virginia, I had to include our lovely copperhead as well as our cottonmouth. Copperheads are found throughout the entire state, whereas cottonmouths are only found in the eastern portion of the state. Our cottonmouth is Agkistrodon contortrix, which means Agkistrodon is fish hook fangs. They actually share the same genus. Contortrix, actually, I had to learn this recently, is more related to their patterning. So you can see the beautiful, intricate patterning that's very twisted and kind of shapes. A lot of people usually associate it with being a Hershey Kiss shape, but the twisted kind of intricate patterning of that copperhead gave it its name of the contortrix. Agkistrodon fishhook fangs of the piscivorus, which is our cottonmouth, is a fish devourer. They're actually known to eat fish. Cottonmouths also live usually in kind of swampy, river, lake-ish sort of environments. They're beautifully floating as they're slithering through the water, but are known to eat fish, and they got their name justifiably so. So overall, sometimes the coloration, the patterning, but also the behavior can be associated with the animal and what its name is given to it. A few other examples is our narrow-mouthed toad, which is Gastrophene carolinensis. Gastrophene is the belly toad. You can see it kind of lays on its belly there. This guy was found near Danville. But carolinensis is named because it was found in the Carolinas, specifically South Carolina. So sometimes the location of where that animal is found can be used for its scientific name. Now I want that one. Whereas the green frog, Lithobates, which means stone, walk, or haunts, Clamitans, Lithobates, stone, litho meaning stone, bates meaning haunt or walk, is usually because of the fact that sometimes they're not quite this green. Sometimes they're kind of a brownish color, so a lot of times it was kind of like, oh, it's a stone frog. So it got its name with that. They can have variation in their coloration. And then Clamitans, because they're loud calling, because they are obnoxious. They're super loud, and it's one of those, like, you be walking, and you just, all of a sudden, it's like, well, there you are. Whereas some of these other guys would kind of keep quiet and be able to move on. With their common names, I got a little over there, a narrow-mouthed toad, you can see his little cute face there. He's got a very narrow nose, and then this one's kind of blatantly obvious, at least with the green frog, although they can be kind of brownish sometimes. So overall behavior, location, coloration, and physical appearance can all contribute towards the naming of something scientifically, but also commonly. The common name being the one that we use within common tongue, whereas the scientific name is more commonly used within scientific publications, and obviously people that are more interested in the Latin and understanding the specific ecology of that animal. To give you context, this is a little hard to look at. I apologize, it's kind of I didn't want to make my own, but you are here. So, to give you context, this is the entire phylogenetic tree of everything. It's a lot. You are, so I took a little blow up of that you are here that's all the way up there to make it over here. With that you are here, you're just one of those little, honestly, it's a blob. Oh, hold on, I have to do it this way. With our phylogenetic tree, so we have our kingdom, we are specifically within Animalia. We are animals. Phylum Chordata, we have a chordate. We are chordates, we have a backbone. Class Mammalia, we are mammals. I'll let you guys Google these terms later. Within order, we are primates. Family, Hominidae. Genus, Homo, and then species being Homo sapiens. There are potential for subspecies, but we're not going to go into that with humans because so far our genetics only go as far as Homo sapiens. There are also various levels of taxonomy that can be super this, sub that. There's clades, there's tribes, there's all these different levels of taxonomy, but a lot of it gets down to you have these kind of overall building blocks, and then you have these little kind of almost caveats in between, depending on how specific certain people want to get into the taxonomy of that group. Certain people are what are called splitters, certain people are called what are called lumpers. So it's kind of a generalized term, but the overall idea that if something has just slightly different aspects than the other ones, sometimes it gets put into a different group. If it has less common things, sometimes you can get those supra and sub aspects within their taxonomic levels, and sometimes eventually it might become a whole new branch, and you can end up being on a completely different chunk of this wheel. I like this version because of the fact that it puts everybody kind of all around, whether they're still extant or extinct. To give you context, though, if you see fungi is down here, bacteria and archaea are these little ones down here, protista, and then plants, and then animals is only so much of this. So we are only part of this circle. One other example related to our naming is the ring-necked snake that we have throughout most of eastern United States. There is a northern and a southern. We're actually within Virginia and specifically within parts of the Appalachian region, especially down where we are in Henry County, part of that integrated zone where they both could potentially occur. And sometimes this isn't always perfect. Sometimes the ring can be broken or connected, but we'll get into that in just a second. So Diodophis punctatus is the genus and species. Diodophis meaning the headbanded snake, kind of blatantly obvious. Has anybody seen a ring-necked snake before? You can raise your hand if you want. A few of you. They're cute little guys. I love them. Find them all the time. Beautiful orange coloration on their belly. One of the most easy to kind of teach about snakes because of the fact that it's no thicker than your pinky. They're just long little dudes. Love them. Punctatus is the spotting that occurs on the ventral side. I should have included a picture of that, but overall gives them the species name. But if you notice, we also have Edward's eye. Edward's eye was actually an ornithologist. I had to Google this. I didn't know it offhand. George Edwards was a ornithologist that lived many, many years ago and was known for kind of writing descriptions, making scientific drawings of certain species of animals, certain species of plants, and was kind of a honor to him when this was described and gave the name to the, I believe, the southern population of, no, it's the northern, I apologize. It's the northern population that is Punctatus edwardsi. The southern population, to help with the separation of those two, because of their geographical separation, Punctatus is the southern. With me so far? Okay. Hopefully, wrong way. So, overall, say we find an animal that we're going to say we have a turtle that we find out in the woods. Everybody loves turtles. They're really cute. Honestly, we have a couple of them at the museum, Speedy and Sheldon. Little shout out. So you go to where you are in your locations, but overall you find this turtle and you're like, where is this turtle from? Is it this species? Is it that species? Is it a new species potentially? Where you want to start is do your homework. Where did you find that turtle? Maybe across Maryland, Virginia, and West Virginia. You found it in multiple locations. Maybe it's a different habitat. Maybe it's a different situation. Something about the seasonality just doesn't work, and you're like, something's wrong here. Looking at collections, which we'll get into, this is a screenshot of our database system. It's honestly just a big Excel spreadsheet that I just downloaded from the database. But looking at records that you might find elsewhere, also another shout out to the Virginia Herpetological Society, with being able to kind of follow that knowledge. Like, is this animal in the locations that we found, or is it not? And then ultimately looking at specimens to understand where we were. Within field work, sometimes you don't always find what you're looking for. In fact, this is a video that we were hoping to catch a beaver on. Can anybody guess what that is? It's a coyote. It's a very strange beaver. Active research, hoping to find beavers in Henry County for other research. But mostly I wanted to give you an idea of what it's like to do active research. Sometimes you can easily find that turtle, put out a turtle trap, find a turtle, take some pictures, get some really good shots of it. Sometimes you're searching for it, and sometimes you really just find everything else from what you're looking for. Remember the snake that we found earlier? I found it with the mammalogy curator. Usually everything kind of happens on accident, or sometimes just happenstance, and that's some of the best science in my opinion. I love that coyote, it's really fun. So, once you found your turtle, yes, I placated that on purpose because it's a turtle picture, but this is a beautiful little snapping turtle. Check the collections. So where I come in as the technician is a visiting researcher that can come to the Virginia Museum of Natural History, can take their specimen that they found and be like, hey, I found this turtle, what do you think it is? Or do you have more of it from those locations, something similar? We can run all sorts of different aspects to it. So we check the collections, we check the collections, we check the collections, and we check the collections. We have all sorts of collections that run all the different types of overall possible preservation types, but we'll get into that in just a minute, and basically going around and comparing everything that we have within our museum, something that's specific to Virginia, and being able to see, is this turtle that we found the same one that we have potentially in our collections? Within this example, we do in fact have a snapping turtle. This is a common snapping turtle. We have it in dry collections, we have it in wet collections, we have DNA, tissue samples, things like that, all that we can use to compare to that specimen that you found to see whether it's a new species. If Virginia Museum of Natural History doesn't have it, you might have to go elsewhere or check just other types of collections and kind of work on collaborative research. So for example, some of our types of collections, I mentioned this at the very beginning before I really got into it, we have different aspects of our collections. Sometimes the tissues, which I'll start right here, is the very bottom right. Within our tissue collections, we keep tissues within every specimen that we process because of the fact that sometimes DNA can be more useful than the actual specimen itself, or sometimes you might even have just a piece of that animal and not the whole thing. So you just kind of work with what you got. Other collections we have, these are some of our dry collections. So we have a beautiful collection of squirrels, which is in reference to our retired mammalogy curator. Nancy had a huge, beautiful legacy looking at squirrels across Virginia. We have bird collections that have been donated to us from local universities, bat collections, we have the nest and egg collections, some turtle shells, which is hint, hint, nudge, nudge to our turtle that we talked about earlier. And then occasionally you can have sometimes the overall comparative anatomy looking at some aspects of certain species of reptiles and amphibians and how things compare to each other while still connected all together, still articulated. And then one of my greatest volunteers, Jenna, kind of going through our collections to being able to separate one thing from another, but also separate things from different localities. So she is actually working through our collections that were separated from a whole pile of things within the same jar to being able to have them separated taxonomically, but also geographically. So that way it's an easy reference collection that a visiting researcher can come in and be able to look at that comparison of, I want to look at, say, shrews from a certain county in Virginia and be able to do that. Jenna was very pivotal in that, and I will always be in her debt for that. Looking at other museums, you continue to check collections. It's what we do. You guys might recognize this beautiful place down here, the Virginia Museum of Natural History, but we also have the North Carolina Museum of Sciences, American Museum of Natural History, and then the Smithsonian Natural History Museum in DC. Everybody works together because of the fact that science is a communal group project. Everybody wants to work together and be able to be like, hey, where's your turtles? What are your turtles looking like? Do you have tissues of that turtle from this location? A lot of collections for the longest time went to the Smithsonian because of the fact that the Smithsonian is one of the oldest museums in the North American continent. Excuse me. Since then, a lot of museums have popped up over the years. North Carolina Museum is several years older than the Virginia Museum of Natural History, but even the Virginia Museum of Natural History, we have thousands and thousands and thousands of collection specimens that we can use as a reference, and we collaborate actually quite directly with the North Carolina Museum all the time, and are starting to collaborate even more with the Smithsonian as a Smithsonian affiliate. So what is in a record? So now that you've found your turtle, overall we want to figure out where that was found, what it is, any details about where it was found, the specific date you found it, and maybe even the conditions of when you found it, where it was, everything like that. And ultimately, this all goes into our database system. These are handwritten labels. Some can be printed, so our insect or recent inverts collections, printed because of the fact that they are very, very small labels. I'll let Cal talk about that some other time. But overall, getting an idea of the snapshot, because you only collected, say, that turtle at this specific date, at this specific location, on this specific weather conditions, or within the certain, whether it was male or female, whether it was nesting, whether it was, et cetera, et cetera. There's all sorts of details, but capturing all that data is pivotal to our records, to our collections, and the overall sharing of knowledge that happens across all museums, but it is still just a snapshot. The importance of a record is the fact that with that snapshot, you can say that before this time, you didn't have that record. You now have that record until insert certain amount of time here. But if there's ever any aspect of, say, the ecology of that area changes, if that species disappears for some reason, you can say that we had this turtle at this location on this date, and if anything changes, what happened? Was it ecological? Was it natural? Was it unnatural? Or sometimes species show up. So certain species of turtle that were not originally from Virginia are now in Virginia. So understanding where that starting point began is pivotal to understanding the ecology of how that animal interacts with the ecology around it. An example of this that actually I can give that I'm sure you all have heard about is the spotted lanternfly. I got some noises. So spotted lanternflies did not occur in Virginia or certain parts of Virginia up until a certain point. Being able to record that, although there's controversy and there's issues with the presence of that species being here, the understanding of that is still pivotal to the research. We are actively collecting things like that to make sure that we know what is happening, and if for some reason, say something starts to eat spotted lanternflies, I've actually seen spotted lanternflies stuck in spider webs. Sometimes spiders will eat them too. Maybe there's something that can eventually fight off that certain introduction of that different species that was not planned to be there originally or naturally. Types of preservation, so I mentioned a few of these. We have our wet collections, which are everything that's preserved within fluids. So you can have things that are stored in ethanol. It's a type of alcohol preservative that we use to make sure that things don't decay. You can also have things in formalin or glycerol, things like that, that are overall a fluid preservation in jars. It's where I work most of the time. But we also have dry collections. So insects are more commonly found within dry collections, but it's not always commonly, it's not everything that, it's not like cut and dry. There's not, everything's one type of preservation or another. You have certain things that have to be preserved in dry collections, certain things that have to be preserved in fluid, but there's also a gray area between those two. These are a couple of our interns we've had over the last couple of years, looking at some of our dry collections up top and our wet collections down bottom, kind of getting an idea of what we have within our collections, and they were very helpful in documenting everything that we have, because we have so many things to work on that by bringing in interns, we can be able to get an idea of what we have in our collections and how we can contribute to other outside research beyond just the VMNH walls. This is also our tissue collection. So our tissue collection is frozen, -80 freezer. Pivotal for a lot of research in understanding the DNA beyond just the physical attributes of a turtle or whatever you might be researching. But just as pivotal and just as important as that physical turtle being in our collections, its tissues can last that much longer. They can also be used for many years to come for understanding how things are related to each other. And finally, our sharing of knowledge, making sure that we have enough connections. Certain species will have different collections that we kind of share a portion of our collections with their museum to be able to make sure that they have just as much contribution towards our overall sharing of knowledge across the globe. But there's also a possibility. So for example, with North Carolina Museum, we're working to describe new species of salamander. Some of those specimens, will stay at the North Carolina Museum, and some will come to the VMNH. Being able to have that in two different places, because it is a collaborative effort, is very common. So having that shared knowledge, that shared kind of mission to make sure that we contribute towards the overall science of the world is beneficial to everyone. And then finally, we get into publishing. Publishing so that way we can actually have everything be brought to you, be able to be published within journals, shared knowledge for continuing research, and it also creates, again, another snapshot in time for that publication, because of the fact that before that publication, you didn't have the understanding of what that research was going for until that publication came about, and then that publication can contribute to everything moving forward from there. One thing that I didn't cover yet so far is our scientific drawings, which I'm working to understand, but a few just kind of little idea of some of the work that I'm contributing towards slowly as we are speaking is getting into scientific drawings that can be used for publications, either just in general at the museum, but also towards our scientific publications as new things come out. Because of the fact that most of the time, having a picture like that, it is beautiful. That is a cottonmouth from our collections, but that's not going to print very well in black and white. Most of the time, most publications are printed in black and white. Getting color pictures printed within our publications is much more expensive. So by having line drawings that are able to show all the anatomical features of that animal and being able to use that towards the publication, even though it's printed in black and white, is more useful than trying to have a color photo that might not show up as well. And it's like, does it look like this? Does it look like that? This gives a kind of clear back and one way or the other, because you can see different aspects of our skull within the, I believe it's a glaucomys, flying squirrel. Few species that have been described just over the last few years. I won't go into everybody's names, but species are still being described. Places are still being discovered. Our world is a constantly growing area that everybody can describe a new species if they look hard enough, if they do their homework, if they take the time to understand what they need to do. These guys were all described between 2024 and 2025. I had to Google that, honestly. But some are very deep sea things. Some are mammals, which are less commonly found, but still able to be found. A jellyfish, a lizard, different type of ant. All these are being described, and some of these things are even being described at Virginia Museum of Natural History. Our curators within recent inverts are constantly looking to find new species of invertebrates to be described, and it's kind of their bread and butter. It's what they love to do. They love to go find all sorts of things and be able to separate things from one to the other and being able to potentially describe a new species. If any of you are ever interested in working with us, we do a lot of outreach and educational programming. We do bio-blitzes, we do all sorts of kind of going out field work aspects. Sometimes it's more just recreational, as you can see with the kayak, but educational programming, conferences, things like that, sharing of knowledge, going to all sorts of outreach things and being able to work together with Virginia Master Naturalists, with all sorts of local universities, other museums themselves, but also within our own walls doing a lot of things like that. We have our festivals that we have yearly. Every few months we have a festival that always talks about the new things that are coming up. We just had a few new things pop up at our most recent Dino Fest. So if you're interested in working with us, I would love to have you. I would love to have you work with us and be able to kind of look at our collections and get a better idea of what we have behind the scenes. Thank you.
All righty, we have time for questions from Marshall, if anyone would like to jump right in. And we ask that you wait until you get a microphone so the people online can hear us.
I'll also repeat it if need be. I apologize if I rambled.
I was just wondering, what was your favorite specimen that you've ever come in contact with in the museum?
Oh gosh, that one's a fun one. So recently, I have a few, if that's okay. One of my most favorite ones that I came into contact with, actually courtesy of Dr. Kuiper and I, kind of searching around in our bird collection, some of our oldest specimens that we have at the museum were donated to us from Virginia Tech. They were from 1845? Yeah, 1845, 1846. Specimen-wise that are just really cool. I am fond of the squirrels. I blame Nancy, but like in the best way. She was our previous mammalogy curator. She had such a phenomenal collection of mammalogy specimens. We have like all the squirrels. It's kind of really neat to be able to see the variation within fox squirrels across Virginia. We have such strong variation in their coloration that from the East Coast all the way to the mountains and Appalachia that are variation, and just seeing that all within the same species is just really neat.
What's your favorite animal to like go out and look for?
Snakes. I have to say snakes. Not gonna lie, when I found that rattlesnake that was in the first slide, I was over the moon. Funny enough, again, I was out with Nancy. We were doing mammals research for small mammals, and she was just parking her car, and it was just right along a little patch of pine trees, and I was like, stop! And she was, what's going on? It's like, there's a snake! And I go outside, and there's a rattlesnake, and I was like, oh, this is so cool! Like, I'm a little partial to venomous snakes because of the fact that everybody always misunderstands them. We have a resident copperhead. Her name is Cabbage Patch. That is at the Virginia Museum of Natural History. I take care of her every day, well, every week. She gets fresh water, she gets fresh mice. I just gave her fresh bedding the other day. And venomous snakes get a lot of bad reputation for being this scary thing that's going to come after you. And it's like, no, that snake is just like sitting there like, why are you going after my water dish? Why are you going after my bedding? And it's like, because you pooped in the corner. I got to clean up the poop because you pooped in the corner. And she's just like, really? I love that snake. I'm like, sorry, it's weird. I know, but I love my job.
So I have a question. When somebody finds something new and it hasn't been discovered before, who gets to name it? Is it always a university, a museum, or can you or I make the name?
Kind of yes, all above. Usually the person that discovers it initially gets to kind of a first pass at describing it. Usually it's a collaborative effort in order to make sure that we have kind of respectful aspects of that, make sure that it is usually related as much as possible to the actual ecology of the animal. If you want to, you can technically name it sort of anything you would like. A lot of times people will do the attributed to another scientist, like with the Edwards eye, being able to describe it after that. I hoped, although I don't really work in recent inverts, if I ever get the opportunity, my career was started as an internship at the Smithsonian years and years ago, working with isopods. And if I ever get the chance to name an isopod, I will name it after my advisor because she literally started my career. So as an honor to her, I would love to do that. Her name is Marilyn Schott. She was a phenomenal person. She is passed, unfortunately, but I would love to describe a species after her. So you can, as long as it's not already named, as long as that name has already not been used, essentially, then most of the time you can kind of get that, yep, I want to call it that, and you can call it that. There's usually a review aspect to it, and make sure that's like, one, appropriate, and two, that it's kind of approved by usually a committee sort of group. I hope to describe a species someday. It'd be really cool, just in general.
Hi, thanks for being here.
Thank you.
I know you mentioned like visiting research ships and then also sharing specimens between different museums. Is it if you wanted to go study a collection that was at the Illinois museum that you mentioned, is it easier to move researchers or move specimens?
Easier to move researchers. My preference, if I were to ever have anybody that wants to work on our specimens, is them come to us, because then I don't have to worry about the specimens being lost in the mail. Or I can also keep track of what's happening. Not that I don't trust the person coming in, but it's a matter of, if it stays where it is, then that's a much just easier logistics. When I was previously at the Smithsonian, we had incoming researchers all the time that would, instead of sending them to Brazil or sending them to the UK, they would come to us. We can house them, because people are easy to house and take care of, because you just feed them and they're good to go. And you can also make that collaborative connection with, oh, you've worked on this. And my favorite thing about it also is the almost side projects that can come out of that. So actually, Dr. Kuiper and I were talking about this earlier. I have a huge fish collection that was donated to the museum recently that I'm not a fish biologist. I love fish. They're really neat, but I am not a fish expert. And so I'm hoping that somebody would come to the museum to work on Project A, whatever that might be. I'd be like, hey, can you come identify these for me? Because I don't really know what they are. I mean, I know what a flounder looks like, and I know like the occasional perch or something like that, but it's like, I got a whole pile of fish that I'm just like, please identify. So sometimes you can get the side projects that are just overall beneficial to both, but also kind of create that better collaborative connection that is more personal. Anybody else?
I'm trying to form the question here.
No worries.
What is the minimum size? I'm sure this is different with different species and things, but how do you differentiate between a species that you've discovered and just the individual variation that you may have in that creature? What size, or where do you make that, draw that line?
There's a lot of debate with that line, actually. It's an obnoxiously large gray area, and it depends from scientist to scientist. Because sometimes, like our narrow-mouthed toad that we had earlier, we actually just did a BioBlitz recently with the Virginia Herpetological Society, where we found narrow-mouthed toads that were almost like rust orange. Looked very different from that individual that you guys saw earlier that was kind of like a palish pink to brown coloration, but they're still considered the same species. So sometimes people will see that and be like, oh, it's a different species, but you can have different color variation across a lot of things that are all still the same species. A lot of times that can come, some of the ways that you can kind of come to conclusion on that is the DNA, is the tissuing, and that's more of the modern age style of being able to definitely differentiate between things. But there's actually a whole aspect of being able to describe a species where the geographical variation, can they physically get to each other? But also gametic, so they're gametes, sex cells, that whether they are compatible, can they breed? Also seasonality, can they physically find each other during the breeding season in order for those gametes to even find each other? There's 2 or 3 that I'm missing and I apologize, but there's about 6 or so different variations in how these things can potentially be related to each other or not related to each other. And most of the time, if they can still interbreed and create viable offspring, they are considered the same species, or at least sister species. And then sometimes, eventually, you can have the separation of that geography, or the separation of the gametes, eventually separation seasonality that might potentially cause them to become more speciose and be separated from each other. But there's still some fine lines of, well, what if they come back to each other over there instead of over here? So it's kind of an ever-growing thing, but that's kind of the excitement of the biology, especially related to taxonomy, is the fact that we're constantly learning more and more. But yeah, DNA usually helps a lot with that. Kind of like, this is definitely that, or this is definitely that.
We're getting on an hour, but we have time for maybe 1 or 2 more questions if there are any out there.
If anybody hasn't been to the museum yet, please come visit.
I'll underscore that. So with that, I will just say that I know that Marshall will be happy to stick around for one-on-one questions. I want to thank you all so much for coming out tonight. I do believe I'm actually the next speaker, so I will be here. I believe it's October 21st. Jenna, wherever you are, does that sound right? Okay. Well, anyway, I think it is, but it'll be on the website. It'll be just fine. So please come out, and I'm going to be talking also about collections, but from a different standpoint and different kinds of creatures. So please come out for that. Again, I want to thank the Wayne Theater, the South River Watershed Coalition. I want to thank all of you here tonight and everyone online. And let's one more time, please give Marshall a warm round of applause to say thank you.
Thank you.